I sat in the small, dark cubicle while I waited for the phlebotomist to try to take my blood again. Again she stabbed me, again she missed. Eventually the head phlebotomist came over to finish the job. I was at the National Institute of Health, NIH, in Bethesda, Maryland, serving as the control sibling for my brother. A control sibling is a non-affected sibling of the patient. They are used to compare blood work and other test results to see where the affected sibling differs from the non-affected sibling. My brother has mitochondrial disease, mito, and hemophagocytic lymphohistiocytosis, HLH. Mito is a genetic disease that occurs when the mitochondria do not produce enough energy for the body to function properly. It can affect almost every part of the body. Mito can be inherited from parents. HLH is a rare inflammatory disease that is usually found in infants and young children. With HLH, the cells of the immune system become overactive, causing inflammation. The white blood cells attack all other blood cells. The immune system attacks the organs, tissues, and bone marrow, and when activated, HLH becomes a quickly life-threatening disease. It is a miracle that my brother is alive with both mito and HLH, and NIH doctors were curious to learn why he had survived and find the link between mito and immune dysregulation. Never did I think a mouse would matter. Dr. Peter McGuire's mini-study is working to find links between immune system function and intermediary metabolism. This study is the first organized effort to examine immune function in patients with mitochondrial disease. These investigations are complemented by concurrent studies of immune function using mouse models. Immune cells drastically alter their metabolic programming during activation and differentiation. As a family personally impacted by this study, we have seen the impact on clinical care. Since our first trip to NIH in 2018, we have also noticed a better understanding of the role of mitochondria in immune function by the broader medical community. When we arrived in the lab at the NIH Clinical Center, the head researcher showed my family around. The researcher explained to us what they were doing with my and my brother's blood and told us that they were trying to figure out why my brother was alive with his combination of diseases. My blood was used to tell them what was different about his blood cells. The researcher said that they were using our blood to compare us by creating mouse models with our DNA by using our cells. Mice have similar DNA sequences to humans, so they inject our cells into them and run research that researchers wouldn't be able to do on humans. These mouse models are also used to study the relationship between infection and why people with mitochondrial disease get so sick. One of the studies led by Dr. McGuire and his colleagues was published in the Journal of Translational Medicine in 2023 and is titled Inflammatory and Interferon Gene Expression Signatures in Patients with Mitochondrial Disease. For my brother and others living with mitochondrial disease, this study has led to several groundbreaking discoveries since my first trip to NIH in 2018. Mouse models have linked primary mitochondrial dysfunction to systemic inflammation. Mouse models have linked systemic inflammation to increase in serum levels of inflammatory cytokines in the skin and liver. Treatment supporting mitochondrial function affect immune-related gene set expressions. The treatment method found to help patients with mitochondrial disease is called the mito cocktail. Dr. McGuire's study compared mitochondrial disease patients treated with mito cocktail against untreated mitochondrial disease patients. This comparison suggested that patients receiving treatment have positive enrichment of mitochondrial ribosomal and RNA processing gene sets and of NK and T cell gene sets. Mitococktail treatment includes any one or more of these mitochondria-targeted compounds. These are collectively termed mitococktail. Not only have mouse models helped my brother and the entire mitochondrial disease community, going to NIH and being a participant in the research sparked my interest to learn more about how animal research helps with human research. Thank you to these researchers for conducting this study.